You're listening to the European Parliamentary Research Service podcast on organoids.
Ever heard of organoids? Well, pay attention because these artificially grown organs are opening a whole new world of possibilities for treating disease, drug development, and personalised medicine. Curious about it? Stay with us.
What if we could cultivate mini brains to unlock the mysteries of dementia and mental illness, or produce mini guts to advance research on cystic fibrosis or bowel cancer?
Thanks to organoids, everything we have learned from 2D cell culture can now be tested in 3D. But what are organoids exactly?
They're small clusters of human cells that are grown in a lab to create miniaturized, simplified organs that mimic some of the physical functions of real livers, hearts, kidneys, lungs, and many other body parts.
Generated from either resident progenitors in real organs or derived from tissue cells or stem cells, organoids have the same genetic characteristics as the persons from whom they are taken and so respond to drugs in the same way that person would.
Being able to reconstitute organ functions outside the human body opens up almost unlimited possibilities for scientific research.
It does, as organoids are expected not only to advance our knowledge of how our body and organs work, but also help to develop more personalised therapies and regenerative medicine applications and discover new, more effective drugs.
Recently, scientists set up the world's first living biobank to store patients' tumours and use the tissue to identify the most promising drugs for each person's disease.
In not so many years from now, organoids could become part of routine medical practice or might even be transplanted into patients to replace damaged organs. They could also reduce animal testing and experiments on patients.
But despite researchers' best efforts, organoids still fail to replicate important features of real organs, such as blood vessels, nerves and immune cells, or interactions between organs.
So, is there a downside?
Well, along with numerous opportunities, the generalised use of organoids entails safety and ethical risks, as culture methods are still in their infancy. Another concern is that personalised organoids may facilitate the deployment of personalised medical trials, which may in turn pose new risks and affordability concerns.
Storing this type of biological material in biobanks for such a long time also raises questions related to privacy and consent, terms and conditions of use, or ownership of the bodily materials.
The question is, exactly when does a collection of cells become a living being, and what are the ethical implications as we near this point?
It seems that when science develops so fast, ethics can't keep up.
You're listening to the European Parliamentary Research Service podcasts.